short ID table tonight because I wanted to go into a little more detail. These are going to be just a few of the DNA committee's finds, and I thought it would be fun to identify them. But just for my intro slide, does anyone know what that is?
A squishmallow?
That is correct. Thank you very much. Just wanted to make everyone aware that there is a mushroom squishmallow. Squishmallows for people who have to buy a gift for a child, even an adult child. Squishmallows are awesome. People love them, and there is a mushroom squishmallow, so check it out. Yay. All right, so let's see, for our December table, it would be nice if my slides wanted to advance. The first one is an observation by Sheldon Shelley, who is a member of the DNA committee. Sorry, my dog is barking. Can't do anything about that. And this is a mushroom that he found growing on a log, dead hardwood log. And it may look familiar to you all. This is the iNaturalist observation. He saw this in October, but honestly, you could see this at almost any time of the year. Like, I would say it's one of the few that you can count on, even in the middle of the winter. Looking at it closer up, what I notice here is there's the top of it, I'll use my pointer, the top of it, meaning not the fertile surface. So, the spores do not come out of here. This is just the top. It has these kind of concentric rings. There's no stipe or stem. It just kind of attaches to the wood. And then the shape of this, could call it a semicircle or, potentially a little kidney-ish, but more semicircle-ish. And then if you look here, good job, Shelley, showing the underside in the same picture. Love a picture that shows all the features. And the underside here is very smooth looking. I don't see pores at all. I don't see gills, just looks very smooth. So anyone know what genus grows mostly on dead hardwood, although very occasionally on a conifer? Has this shape? It could be a funnel, a fan, semicircle, has these concentric zones that tend to get green with age, but can be beautiful colors like ochre and ochre buff kind of color, brown, reddish, no stem, and then has this really unique fertile surface with no gills or pores. I'm just looking for the genus. Let's see, chat. Yes, I love these. Okay, excellent. Lovely. So we got Sterium, false turkey tail, and several types of Steriums. All true. Excellent. Good job, team. Okay. So false turkey tail, and just looking at the tails of these turkeys, you can kind of see why you might think it's turkey tail. And it does look not unlike Trametes versicolor, which is the true turkey tail. They both have these really pretty kind of concentric, feathery, almost looking surfaces. And here is, if you tried to Google this, if you tried to find this, you would find mushroomexpert.com. You would see Sterium ostrea, or Ostrea, I don't know. But that means basically that is the name that you would see. And this also is the name that probably is in most of your books if you guys have field guides. But breaking news, we have new information about North American Sterium. So this is work by Sarah DeLong-Duhan, who is a wonderful young mycologist, and she has done really cool research on North American Sterium, and she's very active on iNaturalist. I think her name is literally her name. It might just be Sarah Duhan. I'm not sure, but anyway, I can put it in the chat later. And so she did this cool work, basically looking at Sterium and determining that what we call Sterium ostrea here is actually not Sterium ostrea at all. That's not a North American taxon, like we don't have it. And it's actually 3 different species. So basically, there are these 3 historical names that in the past people kind of thought were synonyms for each other, or they thought was the same as Sterium ostrea. Sterium fasciatum, Sterium lobatum, and Sterium subtomentosum. So that's kind of what this says. Headline: Stereum austraea does not occur in Eastern North America. And these are the lookalikes who do. And when we sent off the DNA and got back a nice sequence from it, and then we blasted it, we got pretty clear results. There's always, GenBank always has some not great things in it. So if you look further down, there's some that say Stereum hirsutum, but if you look at like the top, 5 or 6 results here, and look at the percentage coverage, it's like more than 99%, and that's all Stereum lobatum. This is from Sarah's paper. So a lot of the time when they split things, it's kind of like, that's annoying. Is it even something that I can do in the field? Like, can I identify these things in the field? Or is it like what we just found out about the Lycogala slime mold, that it turns out it's 100, maybe 100 species are what we're calling one. And I don't think any of them can be like visually differentiated in the field, but luckily these Stereum species can. So thanks to Sarah's hard work, we know that, so the top is Stereum fasciatum, the middle is Stereum lobatum or lobatum, and then the bottom one is Stereum subtomentosum. So the first question you want to ask when you get something that you would have called Stereum austraea is, how fuzzy are, like, how are they, what do the hairs look like? So the first two have kind of matted felty hairs, and you probably need a hand lens, or you could zoom in on your phone to really see this. Maybe hard to see from far away. Or you're very young and have good eyes, then maybe you're fine. Mine are not. So anyway, the first two you can see, they look fuzzy, but they look kind of matted or felty. But the third one, the subtomentosum, tomentose meaning hairy, it has really long hairs. So that's the first distinguishing feature. So all you have to do is look at the top, and you can immediately, nail it to either one of the two that are more felty or the guy who's hairy. And then if you want to distinguish further, so just to go back, I showed you Stereum fasciatum and Stereum lobatum, both were matted, kind of felty. Now here we have the same ones. When you wet them or injure them, they have a color change on the underside, the fertile surface. So Stereum lobatum will turn yellow and Stereum fasciatum won't. Now, if it's fresh, this is great. If it's not fresh, you can chew on it. And if you chew on it, that'll get it going. So basically, Stereum lobatum should be able to be identified by having, not being long haired, but rather having kind of matted felty hairs and staining yellow. Oops. So for ours, I don't think that we got the staining reaction, because I don't think we tried it. And that's a good plug to, we should probably, try to do those kinds of things when we sequence something, because it would be nice to confirm that. But anyway, so that's the news on Stereum. Now, a similar kind of thing. So this is an observation from Serenella, but actually this is from Sequinota, and I don't know who found the original specimen. But you see these beautiful mushrooms here, and I don't know if you noticed, so they're big, they're golden kind of colored. You can see they have, I see they're depositing rusty spores on each other. I see that they have a ring zone or some partial veil remnants here. The gills are pretty crowded, similarly yellow colored. And then, I don't know, I see here, looks like a little blue. That's what I see. So what genus, and again, I'm only asking you the genus, what genus has large yellow fruiting bodies, yellow gills, rusty, voluminous spores, a ring zone or partial veil remnants, and flesh that turns blue with injury? Excellent. Martin said Gymnopolis, exactly correct. And I chose this observation because I think we were talking about Gymnopolis either at our last meeting or the one before, and we were talking about some of the taxonomy issues. So I thought it would be a good one to talk about today. So here's a paper that basically looks at the Gymnopolis junoneus group. So in the past, if you saw that mushroom in your books, you might have it as Gymnopolis junoneus or Gymnopolis spectabilis. But once again, we don't have those, so they're not here. So this really good paper reports 5 species from this group from North America. And these are the ones that we do have. We say that they're in the junoneus group. We don't have junoneus. It's just to say that they're all in the same kind of group. Gymnopolis luteus, subspectabilis, ventricosus voitkii, I don't know, and speciosissimus, I don't know, it's a mouthful. Here's just pictures of a few of them. So this is Gymnopolis luteus, voitkii, and speciosissimus. I don't know that, I think, well, I don't know, I guess luteus looks kind of different, but anyway, let's look at the key. So they provide a very nice key in this paper. The annoying part of the key is that it seems to require microscopy, but there are a few things that I think we can take out of it that would probably help us. The first one is that it helps to know whether your Gymnopolis is growing on hardwoods or on conifers. And ours, you can't tell in the picture, but I'm pretty sure it was growing on a conifer. And then this thing, I think Mitch already mentioned that it had a very nice odor. Mitch, how would you have described the odor?
Yeah, some people did say it had a bit of an anise smell. It smelled kind of floral to me, but I could see the anise-ness to it. Some people also thought it smelled bitter. So, but that's how everybody has those different reactions to smells. So
yeah, I do get also kind of a floral smell from it, but I think people call it anise. I mean, I agree, there's a lot of subjectivity, but I think the difference is here that the other one that it could be. So we started at 1, we said go to 2. And then 2, you could have either of these kinds of colocystidia. Well, no, thank you. We're not gonna do that. But the choices are either 3 or 4, and 3 is subspectabilis and 4 is luteus. And one of them smells lovely and the other one doesn't have a distinctive odor. So boom, we didn't even need our colocystidia. All we need is the host and the odor, and we nailed it. And that is what it came up as. So when we blasted this one, our results were Gymnopolis luteus. So I'm glad that we had that smell data from Mitch's nose and others.
That's awesome.
That's kind of exciting. It's nice when, you follow the key and you find the right thing and you can confirm your answer. Let's see, how are we for time? I'll just go really quickly through the last one. I guess I'm just trying to be really annoying tonight by taking things that we've previously felt comfortable with and complicating them. But this is one that I found at Sequinoda. And let's see. So I think the key features here, you can't see them in this particular picture, but it had free gills. It had, white gills and white spores. You can see in the picture the presence of the annulus or ring. You can see, well, maybe back here, you can see there's a bulb, so it gets fatter at the bottom. We call that a basal bulb. You can see that the cap is not striated. There are no lines here on the margin of the cap. The cap margin is not appendiculate, meaning it's basically smooth. There aren't chunks of remnants of the vulva hanging from it. And it doesn't, at the bottom, it doesn't have a cup or sac-like vulva. It just has the bulb. There are some pieces of vulval remnants, but there's no cup or sac, as some species in this genus are. And then other things, those are all helpful. So, okay, the first thing is the free gills and white spores and the ring being present, that can take us to what genus? Yeah, Amanita, excellent. And then I want to get to a section. This is a really trickier, harder question, and so it's fine if nobody knows the answer because Amanita sections are annoying. But let's say you do have a ring, you do have a bulb, you don't have an appendiculate margin, and you have no cup or sac-like vulva. Can anyone take us to a section? mean, you also might know what this species is, and then you could backtrack to the section, but section Validae. So section Validae, which is also contains some of the blushers, those guys, they have a ring, they have a bulb, they don't have cup-like vulva or sac-like vulva. And so that's good. And this is not rubescent or pink staining, so we know it's probably not one of the blushers. And it has these yellow universal veil remnants, say, warts, yellow warts. That seems like an important clue to what this could be. And so I think, anyone eyeballing this would probably call this Amanita flavoconia. And if it had been blushing, like turning pink, then we would probably call it Amanita flavorubens. Rubens is good because it's like pink and we're talking about blushing, but not this guy. So this is from a wonderful resource that I wanted to share. Rod Tolos is the basically, certainly North American, but also a world expert on Amanita. And he has an amazing website. It's just really exhaustive and wonderful. And this is his discussion of the Amanita flavoconia species. It's often mistaken for Amanita frostiana. And if you want to feel good about yourself and your difficulty identifying things, if you look at Peck's type collection of Amanita frostiana, which is not even in the same section, there are specimens of Amanita flavoconia in it. So it's a mixed bag. So we can all feel better, even Peck messed up Amanita identification. Some of the species he says to compare to here, Amanita elongata, we do get in this area. Amanita erythrocephala, we also get in this area. And those are kind of lookalikes. But I always thought, okay, that group of kind of flavoconioids, whatever, it's elongata, erythrocephala, and flavoconia. And I'm like, that's cool. I can tell those apart usually. But unfortunately, when we blasted this, we got some good work done by Steve Russell from Indiana. And this is what turned out to match up with a provisional species name, Amanita species flavoconia 3. And what that means is that there are at least 3 lookalike species of flavoconia that have different genetic, just even different ITS. So they probably have other differences too, because usually Amanita, you need multiple genes. But so that's, I guess exciting in the sense that we have an unnamed species here, but it's also frustrating because I think we can still call it Amanita flavoconia, but we have to kind of understand that we're talking more in the broad sense than in the strict sense. So yeah, that's the provisional name, and I'll stop there.
Thanks, Annie.
Sure.
It's fun and a good preview of coming attractions for the DNA presentation coming a little later. But first, before that, Tom McCoy wants to show you some of the pictures that people submitted for the photo contest.